Hi everyone. Before we start, in the previous video I had received good compliments. So I would like to thank every one of you who has subscribed, watched and liked the video. In this video we'll be continuing from where we left off in the last where we had discussed about the type 1 and type 2 hypersensitivity reactions. So we'll complete the topic with type 3 and type 4 hypersensitivity reaction in this video. So let's start with type 3 hypersensitivity reaction. So the first question is, who mediates type 3 hypersensitivity reaction? Type 1 was mediated by IgE, type 2 was mediated by other antibodies, whereas type 3 hypersensitivity reaction is mediated by immune complexes. So it is the antigen antibody complex which mediates type 3 hypersensitivity reaction. Now the question is, from where do we have these antigens? So these antigens could be either exogenous or they could be endogenous. They could be exogenous antigens from bacteria or fungus that present externally, or they could be endogenous, such as the DNA or proteins which are produced by our own body. The next question is whether these immune complexes are soluble or insoluble, because that's an important question to ask. Usually, these immune complexes are degraded by the complement system in our body, but the type 3 hypersensitivity reaction occurs when these immune complexes are present in excess, so that our body cannot clear it off, and they start to get deposited. So where they deposit is influenced by whether they are soluble or insoluble. So these immune complexes could be formed by two mechanisms. Either the antigen should be excessive or the antibodies must be excessive. If the antigens are excessive, the complexes formed are usually small. And when these complexes are small, they are soluble. We know that smaller things are more soluble than larger things in general. What happens when they are soluble? When they are soluble, they can travel easily in the blood, and hence they can get deposited anywhere in the body. So we can say that soluble complexes formed by antigen excess has a possibility for producing a generalized or systemic type 3 hypersensitivity reaction. Compared to that, when antibodies are in excess, the complexes formed will be larger. So larger complexes are formed, and larger complexes, these larger complexes will be insoluble, and hence what happens is that they will produce a local reaction. So a local type 3 reaction will be produced at the site where the antigen enters, or at the site where the antigen is administered. Now let's speak about the pathogenesis. So we have antigen antibody complexes. So this is the antigen antibody complex. Usually they are cleared by the complement system, but when they accumulate in the body, they start to get deposited depending upon whether they're soluble or insoluble. And once they get deposited, they activate three mechanisms. The first mechanism is, of course, the classical pathway of complement. When the classical pathway is activated, we know that it involves all the members of complement from C1 to C9. There are some important some important complement proteins that are involved. The first one is a C3A and C5A. So what do these C3A and C5A do? They are anaphylotoxins. Anaphylotoxins means they can degranulate the mast cell and release the contents of mast cell, leading to vascular permeability and so on. These C3 and C5 are also chemoattractants. So what does that mean? Chemoattractant means they can recruit neutrophils, and that could produce inflammatory response. and these neutrophils cannot usually they can usually phagocytize, but in this condition they will release large number of granules and cause excessive tissue damage. The second pathway that is important for us is the activation of platelet. So platelets are activated. When the platelets are activated, it could lead to damage in blood vessels that could lead to tissue ischemia. And finally, Hageman's factor is activated. What happens when Hageman's factor gets activated? Hageman factor, when it activates, they in turn activate kinins, and kinins will lead to vasodilation. And because of vasodilation, there will be edema as well. So these are what happens in type 3 reaction. So there will be mast cell degranulation, chemoattractants that activates the neutrophils, and then there will be tissue ischemia due to platelet activation and degranulation. Then there will be vasodilation and edema due to activation of Hageman's factor. Now, two types of reactions are there. As I have said, when the complexes are when antibodies are in excess, insoluble complexes are formed, right? So these complexes, they do not have the tendency to travel through the blood and form a systemic reaction. Rather, they produce a localized reaction. So such localized type 3 reactions are what we call as Arthas reactions. So they are a localized type 3 reaction, and by definition, it is the localized area of tissue necrosis formed due to vasculitis. So what happens is that these immune complexes they get deposited, and these deposited complexes they cause vasculitis or inflammation of the blood vessels, and this vasculitis causes tissue necrosis, localized tissue necrosis. This is what happens in Arthas reaction. And coming to systemic reaction. Now, who causes systemic reaction? Systemic reaction will be caused by antibody excess. When the antigens are in excess. So when antigens are excess, soluble complexes, they get distributed and deposited at several organs. So depending on the site where they deposit, we'll have several features. So deposit at the kidneys, what will happen? They will cause glomerulonephritis. When they are deposited at the joints, we get arthritis. Similarly, when they deposit in the blood vessels itself, it will lead to vasculitis. Now, such kind of systemic reactions were earlier seen when foreign serum, such as that of horse, was used for immunization, passive immunization, and all. And such a sickness, this kind of type 3 systemic hypersensitivity reaction, which then developed due to the serum of foreign animals, was called as serum sickness. Nowadays, it's not seen much. So serum sickness is a type of systemic type 3 hypersensitivity reaction. So that's all about type 3 hypersensitivity. Now about type 4 hypersensitivity reaction. The first point is it is a delayed type of hypersensitivity reaction. So all the first three types of hypersensitivity reaction occurs immediately, whereas the type 4 occurs after 2 to 3 days after the exposure of antigen. So it takes 2 to 3 days. It requires 2 to 3 days of time to manifest, and it is mediated using the cell-mediated immunity. What is the pathogenesis? So the pathogenesis involved the antigen is presented by the APC, by APCs, and this is our diagram, and they will present the antigen to the T helper cells. Antigen is presented, and the T helper cells, they get modified. These T helper cells, they get modified to form TDTH cells, that is delayed hypersensitivity cells. And these delayed hypersensitivity cells, so they are derived from either the most of them from the Th1 cells, or they are derived from Th17 cells. They could also be derived from the CD8 T cells. So these three cells would modify themselves to specialize and differentiate into T hypersensitivity cells. And these cells, when subsequent antigen exposure occurs, so when the antigen again comes and binds over these TDH cells, they will release a number of factors. The most important among them is the interferon gamma, and the other include IL-2, interleukin 2, then MCAF, that is monocyte chemotactic activating factors, then IL-3, GMCF, all such interleukins are released. What do they do? Now, each one does a specific job. The interferon alpha activates macrophages. So resting macrophages, they get activated. IL-2 proliferates the TDH cells, and monocyte chemotactic and activating factor, that will cause the migration of monocytes. So monocytes are activated and they migrate and they get converted into macrophages, and the interleukin 3 and GMCSF, they synthesize new monocytes. Now what happens? Now these macrophages that are produced, these macrophages and the monocytes and all the new monocytes that are synthesized, they all differentiate into macrophages. Eventually we will have lots of macrophages, and these macrophages will continue to release cytokines. Several cytokines are released, and these cytokines will cause what? They will cause inflammation. And with continuous inflammation, what will happen is these inflammation will continue, will start to produce a non-specific tissue damage, because the microbe or the pathogen against which such a hypersensitivity reaction has occurred, that pathogen will not get destroyed by this reaction. The pathogen will it will have developed mechanisms to escape the reaction or the cytokines released by the macrophages. So the macrophages continue to release without the pathogen getting killed, and as a result these cytokines will start to act on the host tissues itself, and that will lead to non-specific host tissue damage. This is what is happening in the type 4 hypersensitivity reaction. When this reaction gets continued, what will happen is the macrophages will differentiate itself to form flat cells from their usual shape, and these cells are called the epithelioid cells. And then what happens is some of these epithelioid cells will fuse, one or two of them or three of them will fuse, and they will form large multinucleated giant cells. So these are the multinucleated giant cells. And these cells, they will be surrounded by lymphocytes. So they'll be completely surrounded by a rim of lymphocytes, and the lymphocytes themselves will be surrounded. As a response to the inflammation, there will be proliferation of fibroblasts. So these lymphocytes themselves will be surrounded by fibroblasts. This whole structure, wherein there are epithelioid cells and multinucleated giant cells in the center, surrounded by a rim of lymphocytes, and in a peripheral another rim of fibroblasts. This whole structure is what we call as granuloma, which is a characteristic finding seen in type 4 hypersensitivity reaction. So that's all for this video. Before we end, the examples for type 3, I have said about Arthas reaction. First one, there is the farmer's lung. It's a localized Arthas reaction on the lungs. Then for systemic reaction, serum sickness is one example. For type 4 hypersensitivity reaction, the examples include TB. The granulomas form in TB, leprosy. These are typical examples for type 4 hypersensitivity reaction. And that's it for this video. With that, we conclude the session on hypersensitivity reactions. If you do like our content, subscribe and share, and give more suggestions on the comments so that I could take those videos as topics for my future classes. Thank you.